This is an overview of an article entitled Multigenic Disease and Bilineal Inheritance in Dilated Cardiomyopathy Is Illustrated in Non-Segregating LMNA Pedigrees, published in the July 2018 issue of Circulation, Genomic and Precision Medicine. Dilated cardiomyopathy, or DCM, is typified by progressive ventricular dilatation, impaired function, and heart failure syndrome. Approximately one-third of cases have a positive family history, and in the majority of cases, the mode of inheritance is consistent with a monogenic autosomal disorder with age-dependent penetrance and variable expressivity. However, members of the same family with the same genetic variant can have marked variation in phenotype, from no disease expression, that is, mutation-positive, phenotype-negative, to very severe disease at an earlier age. Some members without the presumed disease-causing genetic variant can also express the phenotype in a form that's atypical for the family. This has generally been thought to be due to extraneous non-genetic variables such as untreated hypertension and alcohol excess. However, taken together, these observations suggest the possibility of other unelucidated effect modifier genes or multiple hits in digenic or trigenic carriers. The authors of the paper under discussion have previously reported on LMNA gene variants identified in 19 pedigrees with familial DCM. In 6 of these families, the LMNA variants did not fully co-segregate with the phenotype, and the families were termed non-segregation families. In this paper, the authors build on the aforementioned cohort by testing the hypothesis that additional genetic variants may contribute to DCM in non-segregation families. Each individual's phenotype was categorized using an ordinal grading scale ranging from 0, that is, no phenotypic evidence of DCM, to 4, meaning DCM with advanced disease. Exome sequences were interrogated for a total of 20 individuals across all 6 non-segregation families. The authors went on to construct a linear mixed model adjusting for age, sex, LMNA variants, and any identified non-LMNA variants identified in other genes known to be involved in DCM. Here's a table with the full catalog of DCM gene variants found in the families. The implicated genes are highlighted in the red box. In 5 families, with a combined total of 88 individuals of non-Hispanic European descent, at least one other potential rare and plausible variant in a known DCM gene was identified. In 3 out of 5 families, bilineal inheritance was clear or presumed, and in the remaining 2 families, bilineal inheritance was possible. The non-LMNA variants were classified as variants of uncertain significance based on American College of Medical Genetics and Genomics criteria, but did meet other potential criteria for disease association, such as occurring in conserved regions of the genes. In the families shown here as an example, pedigree L, non-LMNA variants included missense variants in BAG3, RBM20, and DSP, as well as an early truncating TTN variant. Note that the most severely affected family member actually lacks an LMNA variant, but has the variants in the 4 other genes. In the multivariable linear mixed model, the presence of the additional non-LMNA variants was associated with a more severe phenotype, as judged by either decreased left ventricular ejection fraction or increased left ventricular diameter, highlighted in the red box, after adjustment for age, sex, and the LMNA variants. In conclusion, the authors identified additional potential genetic contributors of DCM in non-segregation families with pathogenic LMNA variants. In 5 out of 19, or 26% of families, multi-gene causation was felt to be likely, which challenges the concept of single-gene Mendelian idiopathic DCM models. In 3 families, there was substantial evidence of bilineal inheritance, while remaining a possibility in the other 2 families. In a multivariable linear mixed model, the presence of additional non-LMNA variants was associated with a more severe DCM phenotype. Together, these data illuminate the complexity of DCM and might explain the wide phenotypic variation seen with the disease, as well as highlighting the potential utility of comprehensive exome sequence analysis.